Good morning.
Good morning,
Good
sir.
morning, sir.
Good morning, sir.
Yes. Okay. So today we have 14 people on board. I think rest of them they are enjoying the Sunday. Fine. So let's begin. So since last couple of days or the week, we had been talking about a term that is the metabolomics, or we have also talked about the metabolic engineering. So what are the things what should be done when you are going for metabolic engineering? The first thing is that you should be able to study the metabolomics. So what is metabolomics? So that is what we have seen, that we have to study the whole system, the system as a whole. What is that? That is what we are supposed to study upon. Fine. So there were different aspects, different angles what we tried to understand. So it could be with respect to the substrate which is entering into the cell with the help of the nutrients, in the form of the nutrients. This substrate will undergo a series of reactions, the so-called metabolic pathways, whereby it might be responsible for increasing the biomass, which could be required for obtaining the energy for the different type of the processes within the cells. Fine. So that's the growth phase what we are talking about, and some of the ingredients which are taken in, they could either be excreted outside the cells, or there are few things which are secreted outside the cells. So all these things which are involved into the phases what I have talked about, means starting from entering of your nutrient, okay, till the output what you get out of from the system. Right. So that is what we had been talking about. Then we have seen the different aspects, like if you want to understand the system, you will have to study it from the genetic level. That is, you should know the genomics, then you should know the transcriptomics, you should know proteomics, you should know the metabolomics, you should know the fluxes between them, that is the fluxomics. And one more thing which could come across, that could be called as an interactomics, that is the interaction between the genes. It could be an interaction between the gene and the gene product. It could be an interaction between the gene product and gene product, etc., etc. Okay. So when you're talking about these kind of interactions, we came across something called as an regulation. Okay, so we saw about the regulatory network, we saw about the network, etc., etc. Right. So all these things, if you study as a whole, that is what we saw, that is the systems biology, will constitute your metabolomics. And if you are aware of these metabolomics, you can construct your own network. And if you know the network, you can think of going for the modification into the network, because if you do the modification, you will come to know what kind of changes are going to happen into the system if you know the network. Okay. And if the changes which you have made, it is not affecting your network, maybe you can say that you are successful in your experiment. Right. So that is what is required for metabolic engineering. Okay. So in today's presentation, where we'll try to see that how we are constructing the uh, network, the metabolic network. So it is nothing but an conclusion from all the things what we have studied up till now. Right. I think I have presented my screen, so I'll go into the presentation, which can be called as at the reconstruction of the metabolic networks. Means you have got the complete information now, and from that information you are now trying to construct your own metabolic networks. So for that, what are the things which are required? So we'll just revise it today with this, and there are few additional things which I will put upon. I think the screen is visible.
Yes, sir.
Yeah.
Yes,
Okay.
sir.
I'm audible. There is no problem into it.
No, sir. Audible.
Okay, fine. So yeah. So biochemical network reconstruction, what is required? So it requires the processes for identifying all reactions that comprise a network. And as we know that these networks, whatever you are talking about, they are all interconnected to each other. They are not independent, right? So that is what we had been studying when we started with the biochemistry, that though you may have the individual pathways which are occurring, but these pathways, they are interconnected to each other. So you can see here in this diagram where you can see that any pathway what you take, it is interconnected with the other pathways. So that kind of complexity is there into the overall metabolism, right? So we have taken that into consideration. Now within this system, what actually happens? There is an intermediary metabolism, means it is the overall metabolism which is taking place inside the cell, which is responsible for production of the energy, and that energy is utilized for building some specific biological structures. Okay, so there you can say that you have both catabolism and anabolism type of the reactions. Now whenever this metabolism is taking place, it will obviously take into consideration all the laws of physics and chemistry, whichever is applicable. Means the chemical reactions, they are obeying all the characteristics of a particular reaction, fine. And this overall metabolism what we have, it is under a very robust type of an regulatory system, right? So it will never happen that the cell will go for excess production of energy, or it could be an excess utilization of the energy, etc, etc. So it's a very highly regulated type of an system which we have already studied about it. So if you want to go for network construction, there are 4 levels which we should take into consideration. So for 4 level functional decomposition of the metabolism, what we should do, and if you have understood that, you can easily go for the network construction. So what are those? So already we have talked about it. We are aware of it, but still we are putting it in some format. Okay, so the level 1 could be your cellular inputs and outputs. So that is what I had been telling you, that there are nutrients which are entering into the cell in the form of a carbon. Okay, and that nutrient could be in the form of the substrate which is entering into the cell. Once it has entered into the cell, as I told you, it will undergo the metabolic reactions. The metabolic reactions are responsible for production of the biomass, which could help and the production of biomass and the energy which will help in the growth of that particular organism, and there will be something which could be excreted outside the cell as a waste, right? So a given system will always have an input and it will have an output. Okay, now when I'm talking about output could be the waste, it could be some of the secreted products, it could be the energy which is being released outside the cell. Okay, so all those things can come under the output what you are trying to consider, right? So this is the level 1 what we saw. Then level 2 means what we are now trying to do, we are trying to go in details of all the things. So level 2 could include the different sectors whereby this substrate is converted into the product. Okay, so again you can see here that substrate, it is entering into the cell, it is undergoing some fueling reactions what we saw. So it could be your glycolysis, the TCA cycle, etc, it could be the beta oxidation, it could be the amino acid degradation, etc, etc. So it will undergo all those particular fueling reactions whereby the energy will be generated. Okay, and that energy which is generated, it could be in the form of ATP, NADPH or NADH. Okay, and along with that energy what you are getting the different precursors. Okay, so there are some intermediate metabolites which are, I think I have given you a list about of around 11 to 12 precursor metabolites are there which are playing a very important role. Okay, so these precursor metabolites along with the energy may be involved into another system which is we call as an biosynthetic reactions, right? And through these biosynthetic reactions you get the building blocks, and these building blocks will be then further converted into the typical structures, the organelles, etc., etc. Okay, and that could be responsible for the growth of that particular organism. Right. So we have the input and output. Now we have gone in detail of it that input will undergo this catabolism, anabolism, which is responsible for the growth. Okay, so that could be your level 2. Now level 3 is what? That now we try to go inside what happens in catabolism. Okay, we already seen that what happens in catabolism, what happens in anabolism. So this catabolism and anabolism, it constitutes what? The pathways. Okay, so we have seen that there are different pathways of the catabolic reaction. There are different pathways of the anabolic reaction, right? So we saw that there is a series of reactions which are occurring within the cell, and times. So the pathways in catabolism could include that the substrate which is being brought inside, it is then hydrolyzed, it is activated by some cofactor, and then it is undergoing the oxidation process or the degradation process where the energy obtained. The same way you can talk about the pathways in the anabolism where energy is utilized. Okay, and from the precursor molecules you are getting the building blocks which are formed, which could be responsible for the synthesis of the macromolecules. Fine. So so at this level, the metabolism, it relies on the basic chemical principles such as stoichiometry and the kinetics. So these things also I told you. Like example, like glucose, it enters, it undergoes glycolysis, converted into acetate, acetate will undergo TCA cycle, it will form NADH and FADH2, which will enter into the ETC, and then you get the ATP, which is being synthesized. So that sequence, what would be made up of many different type of the pathways. So level 3 is what that we saw is the pathways, and then obviously the level 4 will be what? The individual reactions of that particular pathway. So within glycolysis there are 10 reactions, so we are trying to understand each and every reaction of that metabolic pathway, right? So it is at this level we are focusing. Okay, so high throughput data has made it possible. We have many sophisticated instrumentation whereby you can study the individual reaction, and if you can understand it, you can means you can understand those individual reactions at different level, maybe at the genetic level, maybe at the proteomic level, or maybe at the actual reaction level, right? And knowing those particular things can create our own stoichiometric matrices, which I told you. So the matrix which I showed, so that kind of matrices can be generated, and these reactions, it could range in hundreds of metabolites and thousands of chemical reactions. Okay, so within the system where you have the catabolism and anabolism, you have hundreds and hundreds of metabolites which are present, and these metabolites, they are going under thousands and thousands of reactions, right? So we are trying to get the information of each taking place into the cell. Okay, so if you have understood this hierarchy, okay, that will help you to in generation of your metabolic. Okay, so how are the things which we have tried to understood? So you can take any organism, you can take an E. coli, a prokaryotic system, you can take an prokaryotic system, any kind of system you can take. So you can see at the background you an E. coli is being shown. So you take an organism. So the first thing what you can go for is that in genomic study you can go. So like genome annotation you can go. So if you have the complete genome, you can annotate it. Annotation म्हणजे काय मी तुम्हाला सांगितलंय की it is where we are trying to find out the functional genes, right? So genome annotation, it could be for homology or the different type of the bioinformatics tool. You can go for the genome annotation. That is one angle which we can study. Then you can study the biochemistry. So biochemistry means what? That what kind of proteins are present, okay, then what kind of reactions are occurring within the cell, etc., etc. So all those catabolic between them, etc., etc. The regulation, signaling, हे सगळं काय येतं बायोकेमिस्ट्री मध्ये, right? So you can have the protein characterized, etc., etc. Then you have the cell physiology. So in cell physiology, we try to understand the physiological angle. Okay, so it could be the direct or the indirect pathways, which we know that if you change the nutrients, if you change the conditions, what kind of physiological changes will occur within the cell. So that aspect can help us to generate this model for your metabolic, and there are something called as an inferred reactions. There are some indirect type of the reactions which could there. Means suppose you are changing the nutrient, like operons, what I told you, that if you have the glucose, the organism will go lavishly. But if I change from glucose to I say lactose or galactose, so at that time it may have some problem. So it may have to exert some of its energy to express some specific genes so that they can grow in that particular media. Fine. So that kind of some inferred reactions are there which are indirectly affecting your organism. So if you have a knowledge of all these different fields, so like genome annotation, biochemistry, cell physiology, then the inferred reactions, etc., etc., then you can go for the network reconstruction. Okay, and once you have constructed that network with the help of some qualitative and quantitative type of the methods, whatever the data you get. So here you are getting the knowledge. तुम्हाला आठवत असेल एक स्लाइड मी दाखवली होती, that you get the knowledge from these different angles and you get the data from the instrumentation. So combining both of them, you can generate your own model, right? And if you have that model in front of you, you can go for any kind of predictions that what can happen into a cell. That is one thing which can do, or what kind of changes you can perform into that particular system so that you get the things in which you are interested. Fine. So this is how you can go for the metabolic model reconstruction. So if you understood this slide, I think the things which I am trying to explain has reached you. Okay, so it's a very self-explanatory type of an slide which will give you the wholesome idea. So from where do you get this data? So हे पण मी तुम्हाला सांगितलेलं आहे, but still genome annotation, biochemistry, physiology, how can you go for it? So for genome annotation, although I have not details, means because you are going to cover it into bioinformatics part, फक्त इथं तुम्हाला काय माहिती पाहिजे की what do you mean by genome annotation. So there are different algorithms are there are different softwares which are nowadays available, like you can easily go for the open reading frame. Open reading frame मध्ये मग तुम्ही stop codon, start codon, promoter, operator, you get all kind of information. So there is a software called as an there were many traditional methods which were there. So we used to perform the experiment, try to get the sequences, then we tried to find out the sequence homology between them, and then we used to talk about that how much amount of similarity is there between the two different type of the genomes. So that kind of techniques are there. But nowadays we have many innovative methods, you can say, new annotation methods like protein-protein interactions, so it could be, you might have heard about fish, you might have heard about chip, which I told you, that is chromatin immunoprecipitation that I told you. Then you might have heard about East2 hybrid cell. These are the some of the techniques which has helped us to understand the genes and genes, or the gene and the gene product interaction. So you can understand the correlated mRNA expression levels, like with the invention of the sequencing what you have. So you might have heard about some pyro sequencing, then ion Toron technique is there. So there are many new techniques in next generation sequencing where the transcriptomic information can be obtained. So whereby you can easily find out that which gene is expressed under what type of condition. So that kind of studies can be there. And then you can go for phylogenetic profile clustering. Okay, so through bioinformatics you can go for it. Then there are some protein fusion experiments which can be done. Then you can go for the gene neighbors, that is operon clustering. Okay, that how operon it works, what are the different type of the genes which are present it. Okay, so that kind of thing. Then there are many automated techniques which are there. So it could include your microarrays. So you have the DNA microarray, protein microarray, RNA microarray, etc., etc. right? So that way you can go for the genome annotation. And the whole information which has been obtained till now, it has been now deposited in some specific databases. So there are many databases which may come across. One name which you should be aware of, or which you should keep in mind, that is called as KEGG, KEGG, that is Kyoto Encyclopedia of Genes and Genomes. So here in this database, you have the whole lot of information with respect to genomics, transcriptomics, proteomics, etc., etc. In addition to this, there are many other hundreds of databases which you may come across. So some of them, they might be having the whole kind of information, but some of them, they are purely dedicated to some specific organism. So like CYGD, which I have mentioned here. So you have the complete genomic database for the yeast. Okay. Then you have this genomes on online database. So maybe within this database, you have the different databases for different organisms. Then you have the Swiss Prot, you might have heard about this. You have TIGR. TIGR is what? It is the Institute for Genomic Research. In addition to this, you may come across some databases which are dedicated maybe to Arabidopsis, dedicated to Bacillus, dedicated to E. coli or the human, etc., etc. Right? So from that you can get the ready-made information. The second, what you require information is the biochemical reactions, the biochemical data. And obviously, as we know that these biochemical reactions, they are taking place with the help of the enzymes. So, and we know enzymes are the proteins. So you get the gene information. Okay. If you, yeah, you can have the gene information. From gene information, you can say what kind of protein it will be there. And if it is an enzyme, we can understand what kind of reaction it is catalyzing. Okay, so we have a hell lot of information which is available with respect to the different type of the enzymes, their genes, etc., etc. Okay, so one representative example which I have given here, like GLK is a gene which is responsible for glucokinase, and catalyzing a reaction between ATP and glucose, which is converting it into the glucose 6-phosphate. Right? So this is the topological structure of glucokinase. And as we know that these enzymes, they are represented by an number, okay, and that is called as an enzyme commission number. Right? So this also, I think, so I have told you during the enzymology part, number, it indicates the class of that particular enzyme, then the subclasses, right? So in the next slide, you can see the whole things. Yeah, so you can see here in this where you can explain that means what are these oxidoreductases type of the enzymes. So here we are considering that who is the donor, who is the acceptor, etc., etc. So all those kind of things are taken into consideration, and you can have the EC nomenclature for your enzymes, right? Then you have a huge literature which is available. So there are many books, there are many research articles are there which are available, there are many review articles which are available. So there also, if you scan the Google, you may come across some specific journals which are dedicated to maybe for Arabidopsis, or dedicated to the prokaryotic system, or dedicated to the human, like human genetics. There is one journal. Okay. So that journals are available where the research articles or the review articles can be obtained, or you may have some specific books which are available. Okay, so and these books could be for specific organisms. So like this blue color, what you see, it is for H. pylori. Okay, so the one of the bacteria which is there into your teeth. So so you have a book on that, you have a book on E. coli, salmonella. So these are some representative books which I have given you, and from this book you can get the knowledge. Okay, now what kind of knowledge? Means the whole the system, sorry, the organism itself, you get the classification, you get the biochemical information, you get the physiological information, etc., etc., of this particular organ. Right. Then but there you will have to cross connect. Okay, means you'll have to the cross referencing ज्याला आपण म्हणतो, we'll have to find out whether the information which is given is really true or not, or whether it is a complete information or an incomplete information, right? So you may have you may visit the databases, you may visit the books, the literature. If you visit the KEG website, KEG website मी तुम्हाला सांगितले, it's a database. So in that, for what they have said that in case of this H. pylori, the glucose is converted to glucose 6 phosphate, then to fructose 6 phosphate, but it cannot be converted to FDP. That on the other hand, one of the book, okay, who is talking about the H. pylori. So in that case, what they are saying that the F6P is then further converted into FDP, right? So like this, you can go for the cross examination and try to find out where you are getting the correct information in which you are interested. Then you may have some physiological information or some inferred reactions which you should also take into, like filling up the gaps. Now here, to understand this particular slide, I can give one example about the amino acids. So as you people might be aware of that, there are some amino acids which are called as what essential, and some of them they are called as the non-essential. So I think you know what is the difference between essential and non-essential. So if you are studying a particular system and acid is being utilized, but that amino acid is not produced in that particular organ. Okay, means one of the metabolite which is not produced into in that particular organism, but still that metabolite is being utilized for production of some other compound. So there is a gap here. Okay, so what does it mean? That particular metabolite which is not produced in the organism could have been obtained from the, okay, so such kind of missing what you can say reactions are there. So you will have to find out those particular type of the gaps, and then you may have to fill up that particular gap. Okay, because see, if you try to provide it through as an external source, again you may increase the complexity. Okay, so if you have your wholesome system where everything is produced into the system and that everything is being utilized for some or the other purpose, okay, so that kind of gaps has to be found out, and then you can fill up those particular gaps and then go for the further reactions. Okay, which I have shown it as written as an example here. So the inferred reactions could be the reactions which are not actually found into the system. So you can think of adding those particular reactions. Okay, and one of the thing which can be added is that the transporters are added in many of the organisms, so that there is an easy uptake of a particular nutrient, so that it can be utilized for the growth of that particular organism. Right? So all those things we'll have to examine very, very carefully. And once you are aware of all these things, you can put or convert the whole information in the form of an mathematical representation. Fine. So on one side, you can see here that I have shown the whole lot of metabolic reactions or the pathways which I have shown. So each and every pathway of an stoichiometric matrix. Fine. So I have explained you how to design this matrix. Okay, so here the rows, it indicate the number of metabolites which are what you can say, the columns, they indicate the reactions, that how many reactions are taking place. For example, suppose if I'm targeting only the glycolysis, so within glycolysis I have something 10 to 11 metabolites, and at the same time I may have some around 11 to 12 reactions. So I have that 11 by 12 matrix. But if I'm having, if I'm considering glycolysis and the TCA cycle, if I'm considering glycolysis, pentose phosphate pathway and TCA cycle, if I'm considering the electron transport chain into it, if I'm adding the beta oxidation into it, so like this, if I'm increasing my scope with respect to the different type of the pathways, what is happening here? I'm increasing the number of metabolites, I'm increasing the number of reactions. So जसं तुम्ही त्याला इन्क्रीज करत जाल तुमचा मॅट्रिक्स पण हा I may say that if I'm studying the 1000 metabolites, so and there are something 4000 to 5000 reactions which are involved in it. So I have a matrix of what? 1000 into 5000. Okay, so that huge kind of developed. Right. So with this, so within this network, a matrix can be formed. So we are trying to follow the tools of linear convex analysis, etc. So all those mathematical, statistical, and computational methods are utilized so that you can have your biochemical network which is generated. Right. Now, once you have generated the network, one more thing which you should take into consideration, that is the gene protein reaction association. That is the gene and the gene product interaction. Okay, like few examples which I'll quote here, and that will help you to understand. So not all genes have a one-to-one relationship with their corresponding enzymes or reactions. Okay, suppose I say there are 5 genes. So not necessary that all these 5 genes will be resulting into the 5 gene products. Okay, so there could be n number of variations. So what kind of variation will be there? इथं मी 2 example तुम्हाला सांगतो, that will help. Like one example which I have given, I think it is of fumarate reductase. Okay, so this fumarate reductase is responsible for converting the fumarate into succinate, which is utilizing FADH2, which is converted into FAD. Okay, now this fumarate reductase, it is what you can say expressed by the genes like FRD, fumarate reductase. So it could be FRDC, FRDD. Means for this, for the formation of one enzyme, you are requiring 4 FRDB, FRDC, FRDD. Okay, so because this enzyme, it consists of 4 subunits, and all the 4 subunits are produced by the different genes. Okay, so you have many genes which are catalyzing only one reaction. Means many genes, many gene products, they interact with each other, they form one enzyme, and that enzyme is catalyzing one reaction. Right? So if such kind of thing is there, so obviously it will create more complexity in your network. Okay, so this is one example. Other example could be a reverse of it, that you have only one gene, but that one gene is responsible for a gene product which is catalyzing many different type of the reactions. Okay, so the example could be here TKTA. This is one of the gene. Now this TKTA, it is responsible for maltolase. Okay, so if you people are aware of the oxidative phase of pentose phosphate pathway, where the ribulose 5-phosphate which is formed, it is again converted back to the glucose 6-phosphate. So it is undergo a series of reactions where different carbon skeletons are obtained, and all those reactions are occurring there, they are catalyzed by this class of enzyme which are called as the transketolases or the transaldolases. So it is a product of only one gene. Okay, so one gene, one product, and that one product is responsible for Lakshadya. Okay, so such kind of interactions are there, or associations within the gene products, right? So you can see here 2 reactions which I have shown. So both these reactions, they are catalyzed by this transketolase 1. Okay, जर अजून तुम्हाला माहिती असेल तर you just visit that particular pathway of the oxidative phase of the pentose phosphate pathway. Fine. So at the end, what you can say that we'll have to integrate the omics data. So you'll have to first have the genomic information. From that you get your ORF, that is the genomics. You have the, then from that ORF you can identify a gene. So you can undergo the transcriptomic studies where you try to understand the expression level of that particular gene. From that you get the information about the protein which is being generated. So you get the proteomic information, and if you know the protein, if it is an enzyme it is catalyzing, so from that you can obtain the flux measurement. That is, you can see that how much, what is the rate of reaction, how much substrate is converted into product, etc, etc. So you get this fluxomic information. So it is the combination of all these omic data which is constituting the metabolomics and helping you to generate your metabolic network, right? And if you understood this, you can think of going for modification. So either at the genomic level or the transcriptomic level or the proteomic level or at this fluxomic. You can think of modification. But I may have told you, but then it's not that easy. You'll have to be very, very tricky here, very logical when you are going for integration and designing your own model. So one example which I have quoted here. So I think all of you know what are isozymes. Can anyone tell me what are isozymes? Yes. Patkan sanga. What are isozymes? अवधूत काय असतं आयसोझाइम्स काय असतात? Vidya?
So two different enzyme having same catalytic activity.
That's all. अजून काही?
No, sir.
Nupur? Aishwarya?
Isozymes are enzymes that differ in amino acid sequence and that catalyzes the same reaction.
Okay, the voice was not clear, but still okay. These are the different form of the same enzyme. They are catalyzing the same type of reaction, but they are present in different compartments. Okay, or they are present in the different organs. Now, if you are looking at a system, if or if you have to overexpress or you'll have to suppress this fructose 1,6 bisphosphate or the aldolase, which gene you are going to target? So if I target this B1773, if I try to block this, okay, so think ha block kela mi, so I may not get this product, but I have these two genes, and these two genes are easily producing the, and still the system will work. So even if I'm blocking one gene, the other two genes are there which will fulfill the requirement. So I may not be successful in my experiment in which I am interested. Fine. So you should try to find out whether such kind of isozymes are present or not. Okay. So that is one thing. So ह्या example वरून तुम्हाला ते लक्षात येईल. I will go to the next example, which could be a bit complicated. And it is of a particular enzyme which is playing an important role in the pyruvate metabolism. Now just look at this diagram and tell me that which gene will you target and what kind of effect it will have if you have to suppress that particular gene. Yes. जसं आता इथं पिवळं करून दाखवलेलं आहे. So if you are targeting this reaction, if you have to make some modification into this reaction which is catalyzed by this enzyme, so it could be either suppression or it could be activation. What you will have to do? Which enzyme you are going to target? sorry, which gene you are going to target. Hmm? So if you look at the arrows, you can get the, you can see that arrows are from these 3 different genes. Okay, so even if I'm trying to or block these genes which are producing some other proteins or some other enzymes, and those enzymes also have the ability to catalyze the reaction for this particular. Okay, so such kind of complexity will be there, where there is an interplay of more than one gene for either the synthesis of that particular or for carrying out that particular reaction, etc. तर ह्या ज्या काही गोष्टी आहेत ह्या तुम्हाला लक्षात घ्यायला पाहिजे. Then only you can be who can go for the metabolic engineering. Okay, so like this there are many reconstructed networks which are readily available. तुम्ही म्हणाल सर आता एवढं तुम्ही सांगितलं मग एवढं सगळं आम्हाला करावं लागेल का? नाही, there is no necessity. That there are some ready-made databases for each and every organism which is now available, and within that databases you get the hell lot of information, metabolomics, transcriptomics, interactomics, fluxomics for that organism. Simply you take the data, construct your own model network, okay, or you may get some ready-made network also which is available, and simply you think of what kind of modification you want to do and you can go for your experimentation, right? So this is a table which will give an idea where you can see that there are some networks, reconstructed networks which has been readily available for these different type of the organisms, like it could be the influenza, E. coli, pylori, cerevisiae, etc. etc. असे रेडीमेड नेटवर्क्स तुम्हाला मिळतील. तुम्ही फक्त तो नेटवर्क घ्यायचा, तुम्हाला काय मॉडिफिकेशन करायचं, यू डू इट and you can go for the successful experimentation of metabolic engineering, right? So this is how in a nutshell I have given you the information about the what you can say, the metabolomics, or how to generate or construct your own network, right? So I think you people are clear with this particular aspect of the metabolomics, okay, whereby you can, where it will help you to generate the network. ठीक आहे, आता नेटवर्क तुम्हाला मिळालं. आता पुढचा एक प्रश्न राहतो आपला, that how do you identify the metabolites? okay, कारण की आपल्याला टार्गेट काय आपलं, that we want to increase or decrease the concentration of an metabolite in which you are interested. So for that you should be able to identify qualitative as well as quantitatively that which type of metabolite and how much it is produced by doing a particular type of an modification. Will be our next topic what we are going to start with. So that topic I am starting immediately. So I'll be having another slide. So yeah, is it visible? The metabolic flux balance analysis. Okay, so we have this concept called as a metabolic flux analysis, MFA, which is a very important characteristic which will help us to go for the identification of the type and the number of the metabolites which are present into a given state of an organism, right? आता परत ह्या प्रेझेंटेशन मध्ये तुम्हाला फक्त लॉजिकल थिंकिंग मध्ये समजून घ्यायचंय. आय मे स्किप सम ऑफ द स्लाइड्स because I'm not going in too much of details of it, but I'm trying to give you an overview that what do you mean by this metabolic flux analysis and how you can go for it, what are its applications, right? So yeah, so why measure metabolites? That is a very good question which should be asked here, that why only we are targeting the metabolites? Why is that you cannot target the proteins, or why you cannot target the genes itself? So the reason could be that they are the readout of the underlying molecular network. Okay, because they are the end products. They infer the enzymatic activities. Okay, means their interconversion into each other is helping us to understand that how the enzymatic reactions are occurring, and they are next to any observable phenotype. Or in other words, I can say that this concentration of the metabolites is giving you the phenotypic changes. The same example I'll quote here, which I told you, that if there is a variation into the production of melanin into a given individual, so obviously his or her skin color will be different. Okay, so the metabolite to be the phenotype of that particular organism. Okay, so if such kind of information are obtained from the metabolites, so that could be the reason that you should measure the metabolites instead of going for the genes. Metabolites are the most what you can say uh, are the most effective type of the thing molecule, which will help us to understand the overall system, right? Knowing this, we can say that it's not that easy. Okay, why? Because these metabolites, they are not the victims, but they are the actors. ओके, म्हणजे तुम्ही त्यांना विक्टिम समजू नका की अरे त्यांच्यावरती एन्झायमॅटिक रिॲक्शन झालीच तरच त्यांचं एकमेकांमध्ये कन्वर्जन, बट दे आर दी वन ॲक्च्युअली विच आर गिव्हिंग यू दी फिनोटायपिक चेंज इन टू दी ऑर्गॅनिझम. सो यू कॅन से दॅट दे आर दी ॲक्टर्स, कॅन हॅव इफेक्ट एल्सवेअर. ओके, जसं की इन्शुलिन प्रोड्यूस होतोय कुठेतरी पॅन्क्रियाज मध्ये, but that insulin which is produced into the pancreas, that metabolite is having effect throughout the body. Okay, so that's why the metabolites are very important here, and that is why we should go for measuring these metabolites, right? So there are different techniques or targets are there where you can go for this metabolomic study. So metabolomics, you can say that we detect the unexpected. ज्या गोष्टी आपल्याला दिसत नाही किंवा आपण इमॅजिन करू शकत नाही, त्या गोष्टी आपण इथे शोधण्याचा प्रयत्न करतोय. ओके, सो इट कॅन बी डन इन टू दिस थ्री वे. आयदर यू गो फॉर टार्गेट अनालिसिस, you go for the metabolite profiling, or you may go for the metabolomics. Overall, everything, all the three are telling you that you are measuring the metabolite, but see, the word itself is giving you a different kind of information. So when I'm saying a target analysis, means I'm targeting some one or two metabolite in which I am interested. If I'm saying a metabolite profiling, means I'm simply looking at a particular class of metabolites. So maybe I may say profile of a particular organism, and if I change the environment, what is the again the change into the profile of that alkaloid. So I am targeting only a few what you can say class of metabolites in which I am interested. And when I am talking about metabolomics, it is the all the metabolites, all the possible metabolites which are produced, which are utilized into a given system. Fine. So the study of the metabolites could be done at these three different level, and at the end with एक टाकलेले where you can go for the metabolic fingerprinting. That's a different story. But then if you are working at the first level, the second level, or the third level, there could be variation with respect to your work. So the scope of your work, the accuracy has been found that if you want an accuracy into your research or the work what you are trying to do, obviously you should go for the meta information about each and every metabolite which is being produced into the system. Maybe it could be at a higher concentration, or maybe it could be at a very very low concentration. Fine. But when you're looking for accuracy, म्हणजे तुम्ही सगळेच मेटाबॉलॉमिक स्टडी करता, म्हणजे तुमचा स्कोप ऑफ स्टडी हा काय होतोय, खूप वाईड होऊन जातोय. Okay, so if you want to have a good scope for your study, it should be a reverse case of it. So instead of going for metabolomics, you should go for the target analysis, right? So it depends upon what kind of work you want to do, whether you are targeted, where you want to go for a specific scope in which you are interested, or whether you want an accuracy in your studies. You can think of any one of these method, either target analysis, metabolite profiling, or the metabolomics. So as I told you, metabolomic is and detect the unexpected. So there are many applications which could be thought for these metabolomic studies. Like it can be used as a technique to have the early disease biomarkers. Means you can identify some specific metabolites, and identifying those metabolites can give you an idea whether a person is going to have a particular disease or not. Okay, so that kind of biomarkers could be there. It could happen that in a particular disease condition there is an overproduction of any one of the metabolite, so that metabolite will act as a biomarker. Then it could be used to have a comparative between two different type of the systems. So it could be the mutants or the wild type lines, म्हणजे wild variety आणि mutant variety या दोघांमध्ये जर फरक तुम्हाला बघायचा असेल, so you can go for this metabolomic study. Or if you want to compare between an healthy and a disease tissue, again metabolomics can help you there. Or if you want to see the comparative between the parent and the crosses what you have done, so that comparative can also be done with the help of metabolomics. It can be used for pathway discovery, to understand the functional genomics of that particular system or the organism. Then it can help you to understand the network regulation. Okay, so the network regulation at the genomic level, at the transcript level, at the protein level. Okay, because see, metabolomics मध्ये तुम्हाला काय मिळतंय, तुम्हाला सगळ्या मेटाबोलाइटचं इन्फॉर्मेशन जास्त प्रमाणात तयार होत आहेत, काही कमी प्रमाणात तयार होत आहेत. मग तुम्ही त्याचं परत एन्व्हायरमेंट चेंज केलं तर देट कुड बी अ व्हेरिएशन इन टू इट तयार व्हायला लागले. सो इट इंडिकेट व्हॉट दॅट द एक्सप्रेशन ऑफ द जीन्स इज इंक्रीज, सो द एक्सप्रेशन ऑफ द मेटाबोलाइट्स हॅज इंक्रीज, राईट? और ड्युरिंग अ पर्टिक्युलर डिसीज कंडिशन द एक्सप्रेशन खूप जास्त होत आहे किंवा एक्सप्रेशन, अशा प्रकारे तुम्ही कंपॅरेटिव्ह करून यू गेट द इन्फॉर्मेशन. And you can also understand that why some concentrated type of the actions are there. Ki jasa flowering, if that why at particular condition only the flowering will take place. Okay, or that what kind of nutritional effects could be there onto your system, etc., etc. तर अशा बऱ्याचशा applications साठी metabolomics is going to be a very good tool. Okay, so this you keep in mind as an application of your you're going to study. Although the applications are many, we had been talking about the different type of the methods, so you should not underestimate the complexity what you are going to encounter here. So complexity will be with respect to what? So in the plant system, of compounds. That if I'm talking about secondary metabolite, just imagine primary metabolites is thousands and thousands ahead. That if you are trying to target on the secondary metabolites, just imagine how many compounds you are going to target. So the complexity is with respect to the high dynamic range is there with respect to the concentration or the production of this secondary metabolites. Obviously, this is very important that it has a very diverse chemistry. Okay, and you cannot amplify it. So each and every metabolite is different from each other. So we have a very little knowledge about these metabolites, their pathways, their regulation, etc., etc. Okay, so although आता बऱ्यापैकी आपल्याला माहित झालेले, but still we are not aware of the complete information. So that increases the complexity, and at the end you can say very bad genome annotation is there. That still we say that we have many genomes which are readily available, but still we are not able to annotate those genomes. Okay, means we are not able to find out the functional genes, whether that gene is responsible for a particular protein, etc., etc. So still we are not very good at that particular point. So we have a very bad, very less information about the annotation of the genomes. Okay, so all these factors, they are responsible for the complexity in going for the study of metabolomics. Now, to give an example, I have quoted one specific example with respect to the complexity. Like there was a study which was done in Bacillus subtilis. Soga and his friends, they did and study the Bacillus subtilis. So he tried to find out the different type of the metabolites which are present into this bacillus. So he came across that there are something 1,500 compounds which are present. Capillary electrophoresis and the mass spectroscopy technique, and found that more than 1,500 compounds are present. So if so many compounds are there, so that many proteins or that many enzymes it will require. And if that many enzymes are required, that many genes should be present into that particular Bacillus subtilis. That is what now I can predict here. But actually, when they went for the genomic information, they could annotate the genes only for 650 compounds in that metabolome. म्हणजे 1500 पैकी फक्त 650 मेटाबोलाइट्स साठीचे जीन्स ते शोधू शकले, बाकीचे जीन्स त्यांना त्याच्यात सापडले नाही. Okay, so such kind of complexity is there which should not be underestimated. Okay, so here you can have a direct statement which can be made that the metabolome status the genome status. म्हणजे तुम्हाला जर जीनोम माहिती असेल त्याच्यावरून तुम्ही सांगू नाही शकणार की याच्यात किती मेटाबोलाइट्स आणि किती प्रमाणात आणि कसे असतील. And so for that you have to go for the metabolomics, that is the study of the metabolites as a whole. Okay, so that is why there is an necessity that you should go for the metabolomic studies. म्हणून आपण सुरुवातीला तसंच केलं की why metabolites. Now we can if you are thinking of going for the secondary metabolites, so obviously we know that there is a linkage between the primary and secondary metabolism. So if you are preparing a model for a plant system who is producing a secondary metabolite, so we'll have to consider the primary metabolic pathways also and the additional pathways of your secondary metabolic. And that is why there has to be a link between the primary and secondary metabolism which you should take into consideration. So already I told you what are primary and secondary metabolites or the metabolism and what. So the same thing which I have written here, so I'll not read it out, so etc. So these are the connecting points. So the next question comes that how do you determine this metabolic reaction? Okay, or maybe a question might have come into your mind that how the scientists they came to know that it is the glucose which is converted into glucose 6-phosphate, then fructose 6-phosphate. Now, ha sequence त्यांनी कसा शोधून काढलाय? Okay, so there were many different type of the experiments which were done to understand the metabolic pathways, the sequence of the pathways, the mechanism of the reactions, and the regulatory mechanism. So for all this understanding about the metabolism, there were many experimental approaches which were utilized. So what were those? So the first one could be use of metabolic inhibitors. Okay, so maybe they might what त्यांनी काय केलं असेल, मग ग्लायकोलिस हेक्झोकायनेज ला इनिबिट करणारा एखादा कंपाऊंड घेतला असेल. So they might have put it into the nutrient, so the pathway will be blocked at that particular stage. So they got the information, yes, that glucose when it enters it has to be converted into glucose 6-phosphate, but if the enzyme is inhibited, glucose is not converted, etc. So use of metabolic inhibitors, use of growth studies. Okay, So you can have the growth study of the organism in different conditions by varying the nutrient content, by varying the pH, varying the temperature. So, and from that growth studies, you can now infer what kind of changes are happening into the organisms. Okay, and the third one, what you can use is the use of biochemical genetics. Okay, so you try to suppress a particular gene, you try to overexpress a particular gene, and with that particular type of the genetic modifications, you can study the metabolism, right? Uh, Quoted that A is converted to B is converted to C is converted to D. So in this case, so if I'm supplying A and B, will the organism grow? No, because A and B is not producing the D which is required for the growth. So in this case, if you are simply providing only A or B, there will be no growth or there will be no production of the D. Okay. But if you supply C, directly from C, you can get the D. So here you can see that yes, there is a growth and you can have the production of the D and the growth will be there and the changes can be fine. So he very खूपच simple है where I have considered linear reaction. But if just imagine we have hundreds of pathways having, so there are many hundreds of branches which are there which are interconnected to each other. So how you are going to make a change into that? That's a real question. So here by working on such simple equations or reactions, you can find out that which step it is blocked. Okay, and from so what are the different tools which are required? So आतापर्यंत आपण सगळे tools बद्दल बोललेलो आहोत. So as I have told, and optimization of the condition which is required. So for that, what you require the mass balance, you require the stoichiometric coefficients, etc., etc. Okay, and if you, and this can be calculated from the metabolic flux distribution. Then you should know the reaction kinetics, the concentration of your reactants, and this can be done with the metabolic control analysis. That is the regulatory network which I have told you. Along with these two characteristic flux distribution and the control analysis, you will require an information about the metabolic pathway maps तुम्हाला माहिती पाहिजे. Then you should be able to study the kinetics and the stoichiometry of each and every pathway, each and every reaction within the pathway. Then you should be able to understand the technological constraint. The technological constraint म्हणजे काय, की तुम्ही जे स्टडी करताय ते स्टडी करण्यासाठी तुमच्याकडे पुरेसं इन्स्ट्रुमेंटेशन आहे का, आणि जे काय अवेलेबल इन्स्ट्रुमेंटेशन असेल ते इन्स्ट्रुमेंटेशन तुम्हाला सगळी आणि करेक्ट माहिती देते आहे की नाही. So that particular type of the technological constraints also you should take into consideration. ओके, नाही तर तुम्ही करत चालले एक्सपेरिमेंट आणि तुम्हाला फक्त 50 ते 60% इन्फॉर्मेशन मिळते आणि यू आर प्रोसिडिंग, इट्स नॉट गोइंग टू हेल्प यू आउट बिकॉज यू आर मिसिंग that particular 40% information. And yes, obviously this which is very important, that is the biological. Okay, see the prokaryotic system and that too the bacteria, they are highly moody. They're highly moody. At times they may show a very good what you can say growth, and at times they may not show the growth. Okay, so that kind of biological constraints are also the different theoretical or the computational approaches which we have already studied, which has to be taken into consideration if you are thinking of metabolic engineering. So there are different methods which has been used, and these they are based on the property of your metabolites. Okay, so the different characteristic, the chemical, physical characteristic of these metabolites has helped us to use the different type of the instrumentation. So it could be the shape, size, mass, the charges, etc., etc. So ह्या सगळ्या characteristics वरून आपण वेगवेगळे techniques इथं वापरलेले आहेत, so that you can go for the metabolite detection. For example, mass. We have a technique called as mass spectrophotometry. then you have the different type of the spectroscopy. Using the UV spectroscopy means you are trying to look upon the absorbance, means you are trying to look at what? The electronic excitation. Okay, so the excitation within the electrons. So if you are using the infrared spectroscopy, time you are trying to understand the rotation and vibration within the electrons. हे बघा, UV visible मध्ये आपण excitation बघतोय, infrared मध्ये आपण rotation आणि vibration वर study करतोय. If you are using the fluorescence spectroscopy, we are working on what? The emission phenomenon. वरचे दोन जे होते ते absorption phenomenon होते आणि हा जो आहे तो emissions phenomenon. If you are using NMR or ESR, there we are looking at the spin. Okay, so you might be aware of that the electron they spin around themselves, or the protons they spin around themselves. So we are taking into consideration that spin around the nucleus also. So that is the NMR what we use. Then we can use the gas chromatography. So if your compound is volatile, so chromatography. If your compound is hydrophobic in nature, we use the reverse phase liquid chromatography. RP म्हणजे काय? Reverse phase. Okay, reverse phase liquid chromatography. If you are aware of the charge of your molecule, you can use capillary electrophoresis, you can use ion exchange chromatography. If you know the size of your molecule, you can use the size exclusion chromatography. If you want to study the protein folding, protein binding studies, so in that case we are using some specific techniques called as an FRET, fluorescence resonance energy transfer. FRET, fluorescence resonance energy transfer. Okay, so you can go for the in vivo imaging. Okay, so nowadays many advanced technologies are there. In vivo म्हणजे काय? Within the system तुम्ही फक्त एखाद्या उंदरामध्ये एक फ्लोरोसेंटली लेबल कंपाऊंड इंजेक्शन द्या आणि त्या उंदराला तुम्ही मायक्रोस्कोपच्या खाली ठेवा. ओके, so which is having fluorescence. So you can see that how that molecule is moving within the mouse. Okay, it is converted into which type of compound. So that kind of techniques are available. Protein binding studies जर तुम्हाला करायचे तर histochemistry, you can use the histochemistry, you can use the antibody study, that ELISA, RIA, असे भरपूर ज्या टेक्निक्स आहेत तुम्ही ते वापरू शकता. Right? So using this different type of the technique, you can get the information about your metabolite. आणि याच्यावरून एक लक्षात येते की that there is no single technique which is available which will give you the wholesome information about a particular metabolite. Okay, so if I am interested in an X metabolite, I'll have to do all these techniques. आणि ह्या सगळ्या टेक्निक मधला डेटा घेऊन मी नंतर प्रेडिक्ट करू शकतो that whether then I can predict that what is the structure, what are the characteristic of a particular metabolite. Okay, so compulsorily what use all these type of the techniques for the metabolite detection. So just imagine that how much time you will require. जर ह्या सगळ्या टेक्निक तुम्हाला करायच्या आहेत आणि there are thousands of metabolites, so how much time you are going to require it, right? But still scientists have found some options for it. Okay, so they went for something called as an orthogonal techniques, or one word which I might have used in the initial lecture, that is hyphenation. So what they did, they tried to combine these different techniques. Okay, and by combining those different techniques, what they could achieve the information at a very fast rate. Okay, means doing only one experiment, you are getting 2-3 different type of the information. Okay, so they might have combined the electrophoresis and microscopy. They might have combined not microscopy, but they might have combined electrophoresis and spectroscopy. They might have combined the microscopy and the spectroscopy, etc., etc. Right? So those kind of techniques where we are combining the different techniques, they are called as what? The orthogonal techniques or the hyphenated techniques. लक्षात घ्या. Okay, so you will have to combine, or you'll have to use those kind of techniques which can physically resolve your compound. So each and every metabolite or each and every molecule in which you are interested, they should be properly resolved. Resolved म्हणजे काय? Separate. Okay, your technique should be universal. काही टेक्निक वापरणार आहे ती सगळ्या मेटाबोलाइट साठी वापरली गेली पाहिजे. इट शुड बी सिलेक्टिव्ह. Okay, so it should be able to give you the separation of each and every individual molecule, and it should be sensitive, that it should be able to detect even the nanomolar concentration of the compound, right? So when you are thinking of a technique to be used, these two characters which you have to take into consideration. First one is what? That they should be able to physically resolve your compounds. It should be an universal technique, it should be a selective technique, and it should be a sensitive technique. So you can use fractionation, purification, chromatography, electrophoresis. These are not used for metabolomics, and these are not universal. So त्याप्रमाणे तुम्ही decide करू शकता. Detection साठी you can go for NMR, but when you're going for NMR, it becomes highly selective. कारण की फक्त आपण तिथं protons किती तेच मोजू शकतो. You cannot get any other kind of information. Okay, and in that case, it may have low resolution and low sensitivity. Mass spectroscopy. आता तुम्हाला वापरायचा असेल, so you should be able to ionize the metabolites, but all the metabolites cannot be ionized. So again, it becomes a problem. So हे काही मी तुम्हाला advantages and disadvantages सांगण्याचा प्रयत्न करतोय, that such kind of difficulties are there. Each and every technique will have its own advantages and disadvantages, and according to that, you will have to work upon, collect the data or all the experimentation, and then you can tell what type of metabolites are present in it. Fine. So obviously, this out of these four, which one we can think of? So the last one, that is, you should have a technique which will be giving you higher accuracy and higher precision. Fine. So हे just एक information साठी मी इथे टाकलेलं आहे, हे असं कोणी तुम्हाला विचारणार नाही. So this is what we are actually trying to achieve with respect to accuracy and precision, right? So you should have a high accuracy and a high precision. Fine. So yeah. कंटाळा आला का रे? Yes, you're getting the things or not?
Yes, sir.
कंटाळा येतोय? फास्ट होतोय, फास्ट होतोय, स्लो होतोय, कळतंय नाही कळतंय, काहीतरी सांगा मला.
कळतंय सर.
नक्की?
Yes, sir.
करायचं का थांबायचं? काय काय? ठीक आहे, बोला ना तुम्ही, काहीतरी बोला म्हणजे मला थोडासा गॅप मिळेल. ओके, हे थोड्याशा या 4-5 स्लाइड्स ते आपण बघू, अँड देन वी कॅन एंड अप द थिंग्स. फाईन. ठीक आहे. आता हे ह्या सगळ्या स्टेप्स आपण ऑलरेडी बघितलेल्या आहेत अबाउट द नेटवर्क कन्स्ट्रक्शन, त्यामुळे आय विल नॉट गो इनटू इट. सो दीज आर द डिफरंट स्टेप्स व्हॉट वी हॅव ऑलरेडी सीन, अँड वी हॅव डेव्हलप्ड आवर नेटवर्क. हे चित्र मला मिळालं होतं. दिस इज अ व्हेरी गुड इमेज विच विल हेल्प यू टू अंडरस्टँड अबाउट द फ्लक्सेस. जसं मी सुरुवात केली होती दरवेळेस की यू हॅव अन इनपुट, यू हॅव अन आउटपुट. ओके, म्हणजे तुमचा इनपुटचा एक फ्लक्स आहे. एकदा इनपुट मध्ये आलं तर विदिन दॅट सिस्टीम, as I told you, that it is undergoing the different type of the reaction, so that you can call it as an metabolic pool. Okay, and at the end the sink fluxes is what? That the output what you are getting. Okay, so you have the source fluxes. So इथे इमॅजिन करा की बघा आता कुठून तरी समुद्रातनं पाणी येतंय धबधब्याच्या इथं. सो दिस हा धबधबा जो आहे दॅट कॅन बी कॉल्ड एज व्हॉट द सोर्स फ्लक्सेस. आणि मध्ये कुठेतरी एक असं तुम्हाला प्लॅट्यू रिजन येईल की ज्याच्यात पाणी थांबून राहतं. सो दॅट कॅन बी कॉल्ड एज व्हॉट द मेटाबोलाइट पूल. ओके, सो इट विल स्टे देअर फॉर अ वाईल अँड देन इट विल गो डाऊन द म्हणजे ते धबधब्याच्या खाली जाईल. सो यू विल हॅव दिस सिंक फ्लक्सेस, राईट. सो ह्या पिक्चरवरून तुम्हाला ते लगेच कोरिलेट करता येईल दॅट व्हॉट आर दिस फ्लक्सेस, व्हॉट काइंड ऑफ फ्लक्सेस could be there. So it could be the source fluxes, the metabolite pool and the sink fluxes, right? So जर तुम्ही प्लांट फिजिओलॉजी, आपण एवढं डिटेल शिकत नाही, पण प्लांट फिजिओलॉजी मध्ये एक कन्सेप्ट आहे सोर्स टू सिंक. Okay, so the transportation of the water and the nutrients within the plant system is called as source to sink. Okay, fine, ते आता डिटेल मध्ये नको जायला. असो, आता मेटाबॉलिक मॉडेलिंग ऑलरेडी केलंय, याच्याबद्दल मी तुम्हालाही बऱ्यापैकी सांगितलेलं आहे. आय थिंक आय टोल्ड यू अबाउट द मॅट्रिक्स विच कॅन बी कन्स्ट्रक्टेड हिअर. So these are the different derivative equations for each and every metabolite. This is how you can put it on, that in which reactions it is involved, whether it is formed or whether it is utilized. Then we have put it into the matrix form. Okay, so this is the larger one is called as what? The stoichiometric matrix, that is S. Okay, and the small what you see is the number of reactions. Okay, so usually we multiply these two matrices, and the product what you get is 0. Okay, so whenever you are getting a product of this S into V, okay, that is the stoichiometric matrix and the reactions. Okay, so if the product is 0, so we call that the system is at a steady state. Okay, that it is a very, very stable type of an system. Okay, so the time derivatives of these intermediates are 0 under the steady state assumption. Okay, so S into V, which is a very good factor which is being used for the different type of the what you can say mathematical or statistical calculations about your metabolic modeling, which is being used. Okay, so it depends upon the product of this S into V, which is going to decide whether your system is an stable system or an unstable system. ठीक आहे, मी मुद्दामून याच्या जास्त डिटेल मध्ये जात नाहीये, फक्त लक्षात ठेवायचं इट इज द प्रॉडक्ट ऑफ स्टॉइशियोमेट्री अँड द नंबर ऑफ रिएक्शन्स, वेअर विच इज गोइंग टू डिसाइड whether it is a steady system or not. So what is this MFA? So the determination of the metabolic fluxes in vivo means how much a particular substrate is formed and how much it is converted into the product. If you can know if you can determine this flux, that particular method is called as what? Metabolic flux analysis. So there are 3 steps which we take into consideration, which are used for the systematic investigation of the metabolic fluxes and their control. So the first one is what? That you development of means to observe metabolic pathways and measure their fluxes. Okay, so means we try to find out some methods, some techniques, okay, where you can observe these metabolic pathways or you can measure these fluxes. Fine. So then you can go for what you can say modification. Okay, so if it's a stable system, you can go for the modification and then again study the metabolic flux. Okay, so पुढे एक अजून सांगतो मी. So ह्या 3 steps आहेत की which you have to follow. So step 1, so development of means to obtain flux measurement still tends to be a problem specific. So still we do not have the accurate methods where you can go for the determination of this or the measurement of this flux, but still radioisomers or the radioactivity has been really helpful in finding out the metabolic fluxes. But nowadays this radioactivity has been replaced by the fluorescence. Okay, because you know what are the disadvantages of the radioactivity, right? So radioactive methods can be used where you can understand the flux measurements. Fine. Then step 2, what we do, we introduce some perturbations. Now, what do you mean by perturbations? Means disturbances into the system. Means either you modify a particular gene, okay, or you change the environment. So any kind of the perturbations can be brought about, and that changes can be brought about by using your genetic engineering techniques, right? So in the step 2, what we do the modifications into the system, and in the step 3 again we go for the flux measurement. Okay, म्हणजे मॉडिफिकेशनच्या आधी आपण एकदा फ्लक्स मेजरमेंट करतो, त्याच्याकरता आपण टेक्निक शोधून काढतो, देन वी गो फॉर द मॉडिफिकेशन, अँड आफ्टर मॉडिफिकेशन वी अगेन ट्राय टू मेजर दी फ्लक्सेस, आणि मग आपण ह्या तिन्ही स्टेपचा जो आहे तो डेटा कम्पेअर करतो, अँड फ्रॉम दॅट यू कॅन सी that what is the metabolic flux analysis which is there, right? So implementation आपण नंतर करतो. So after the key parameters of flux control has been determined, one needs to implement those changes usually by applying the genetic modification. ठीक आहे, ते मी तुम्हाला सांगितलं. So flux analysis, what it helps, what does it do? It organizes the data. So we must define what we require, what kind of data we require. So we'll have to define it. So what are those? So you can see here, when you measure everything that goes in, म्हणजे काय, input. लक्षात घ्या, when you measure everything that goes in means the input. When you measure everything that goes out, म्हणजे output. And you know the metabolic routes to connect this, means all the metabolic pathways, their interconnections and their regulations. Then you can write a complete description of what is happening in the process. Okay, so this is what your metabolic flux analysis is helping us to understand. Ala lakshat? So by doing this different measurements, you put down into it into a form of a equation. Okay, and in that equation we calculate something called as a degree of freedom. So what is this degree of freedom? So degree of freedom, F is equal to what? Number of substrates, what is your input, plus number of products, what is your output, and number of metabolic intermediates included. So throughout that measurement, whatever the different type of the intermediates or the metabolic intermediates which are involved, so you should have a complete information about it, minus number of constraints. Okay, that what kind of difficulties you might have come across, right? So it could be your technological constraint, it could be the biological constraint. So if you have all this information, you can calculate the degree of freedom. So this degree of freedom will have some typical value, and from that value you can predict about the system. So you can see here 3 predictions which I have shown. So if F is more, then the number of independent experimental measurements available, then the system is underdetermined. Now what are these number of independent experimental measurements? That out of this input, output and the metabolic intermediates, which type of metabolites can be easily measured. Okay, so those are the different independent experiments what we are going to do. So if the freedom, or if the F is more than all these experimental measurements what you have done, so in that case we call that system is underdetermined. Then if F is less than number of independent experimental measurements, म्हणजे तुम्ही खूप experiment केले, and as compared to that तुमचा degree of freedom हा खूप कमी आला, so we call that system as what? The overdetermined system. Okay, and if F is equal to the number of independent experimental measurements what you have done, we can say that the system is solved, or the system is at a steady state condition. Okay, and usually whenever you are doing an experiment, always your system is into a steady state condition. Okay, so the metabolic flux analysis will help you to understand whether your system is at steady state condition, whether it is overdetermined, whether it is underdetermined. Okay, and for that you have to go for the different type of the experimentations. So what is this determined system? So the number of degrees of freedom is the number of independent fluxes and is calculated as follows. So you have this equation, where you can calculate the degree of freedom, that is D is equal to N minus K minus M. What are these? So where D is the degree of freedom, N is the number of fluxes, K is the number of constraints, and M is the number of measurable fluxes. Okay, so yeah, so I told you, if the number of degree of freedom is 0, if they are equal, so in that case, so you can say that the fluxes are determined as a unique solution and the system is an determined system, or it is a steady state system. Okay, so D is equal to 0. का D is equal to 0? कारण की एक एक्झाम्पल जे आपण आधी सॉल्व्ह केलं, सो त्याच्यात नंबर ऑफ फ्लक्सेस किती होते? 5 होते. कन्स्ट्रेंट आपण कन्सिडर केले 3 कन्स्ट्रेंट इथं कन्सिडर केले, सो K इज इक्वल टू 3. आणि ह्याच्यातले 5 पैकी फक्त 2च नाही 3 मेटाबोलाइट होते, त्या 3 पैकी फक्त 2च आपण मोजू शकतो. ओके, सो M इज इक्वल टू 2. So 5 minus 3 minus 2, 5 will 0. Okay, so you can vary only this, you can measure only these two variables, that is V1 and V4, and you can draw a diagram. So V1 and V4 is, if it is calculated, you get a straight line which is passing through, which you get a straight line which is of the format that is y is equal to mx plus c. ठीक आहे, तर एवढं याच्याकडे ग्राफ कडे लक्ष देऊ नका, फक्त इथं लक्षात ठेवा that this is your degree of freedom चं हे इक्वेशन आहे, and if your degree of freedom is 0, your system is a determined system. Okay, and we never work in a determined system because you have a very steady state system, and for that you require something called as an underdetermined system. Okay, so in this case the degree of freedom is greater than 0. Okay, so because there is an immense work which can be done where there is a lack of constraints, so you can put n number of constraints here, and if you are increasing the number of constraints, obviously the degree of freedom is greater than 0, and you have this underdetermined system which is useful for the metabolic engineering, right? So I may not read all this, कारण की एवढ्या detail मध्ये नको जायला आपल्याला. So here only one of the variable is measurable. Okay, so n is equal to 5, k is equal to 3, m is equal to 1. आधीच्या केस मध्ये आपण दोन व्हेरिएबल मेजर करत होतो, इथे एकच आहे. So your D is what? Degree of freedom is 1, and that is why you have a system which is an underdetermined system, and this underdetermined system would be a good system where you can go for the metabolic engineering. एवढं फक्त लक्षात ठेवा. Fine, I'll skip on this. I don't want to go in more details of this. Fine, I can skip this also. Don't look at the complexity at the bottom. So whenever you are preparing a network, so you first prepare a metabolic network. Okay, then you try to find out whether it is thermodynamically stable type of a network or not. You try to find out the maximum enzymes which are involved, the capacity of those enzymes. Then you go for the mass balance of the metabolites, and at the end you study the kinetics. So all these things are considered when you are going for the reaction network, and what kind of constraints you may come across. Okay, म्हणजे constraint तुमचं metabolic network तयार करताना येऊ शकतो, किंवा thermodynamically तुमचं stable system नाही मिळणार, किंवा एन्झाईमची कॅपॅसिटी ही कमी जास्त होत राहते, परत मास बॅलन्स हा कमी होतो, की काही ठिकाणी खूप जास्त मेटाबोलाइट वापरला जातो, खूप कमी वापरला जातो, अँड ऑब्वियसली इट इज गोइंग टू अफेक्ट युअर कायनेटिक्स. सो दीज आर सम ऑफ द कन्स्ट्रेंट्स which can affect your reaction network or the metabolic flux analysis. Yeah, just remember there is something called as a metabolic node. A metabolic node, it comes into picture whenever you have a branched pathway. If you are going for metabolic flux analysis, it will be true for a linear pathway. But what happens if there is a branching? Branching is the problem of it, because your metabolite is getting divided into two different metabolites. So in that case, what can be done? So in that case, you can go for something called as an just let me see what it is called as. You can go for the addition of those particular fluxes. ओके. सिम्पली बघा, जास्त डिटेल मध्ये नाही जायचंय, तुम्हाला परत सांगते मी. आय हॅव शोन हिअर वन पाथवे. सो दिस इज A is converted to I. Further I is converted into B, I is converted into C. There is a branching. So what to do in this case? So here what do? We distribute this overall pathway in three linear reactions. म्हणजे A to A I एक, I to B एक दुसरी, आणि I to C एक तिसरी. So for each and every series of reactions in a linear form, we calculate the flux. Okay, आता इथं flux ला J1 नाव दिलंय. So you will have the J1 flux, you will have the J2 flux, you will have the J3 flux, and at the end you will add up this. So J1 is equal to what? J2 plus J3. Okay, so whenever you come across the metabolic node, or whenever there is an branching, at that time we study each of the linear branch, okay, and then we combine those fluxes. Okay, and that is called as the metabolic nodes. Yes, then I told you about the different type of the constraints. So एक आधीची जी डायग्राम होती त्याच्यात you get this pentagonal type of the image. Fine, I'll not go in this detail. परत तुमचं confusion वाढेल, so I can skip on this. So there we come back to this integration of the multi-omic data. So as I told you, genomics, transcriptomics, proteomics, metabolomics, interactomics, or the fluxomics. So all those things you can combine and you can have the overall system. ठीक आहे. I think I'll stop here, कारण की परत तुमचं कन्फ्युजन थोडंसं वाढेल. एक छोटासा पॉइंट आहे, तो उद्याला इथं डिस्क्राईब करेल. So from this we get an idea about what is metabolic flux analysis. So tomorrow we'll talk about how do you use radioactivity for measuring the metabolic flux analysis, and at the end I will give you some applications of the metabolic flux analysis. Fine. So तो एक थोडासा टेक्निकल उद्याचं लेक्चर असणार आहे आपलं, and after that I think I may require some 2 more lectures. So maybe 2 ते 3 lecture मध्ये I'll try to find up. So there I'll try to talk about some successful example where the metabolic engineering has been done and you might have got some good changes into the organism. So that I will talk about. एक माझ्या मते कोणाचं तरी प्रेझेंटेशन आहे ऑन मेटाबॉलिक इंजिनिअरिंग. कोण आहे?
सर मी सोनम.
हा, मग तू काय विचार केलाय? Have you prepared something on it?
येस सर.
काय घेतलंय?
सर त्याचे बेसिक गोल्स वगैरे एक्झाम्पल.
कुठलं एक्झाम्पल घेतलंय?
ट्रिप्टोफॅन सिंथेसिस.
कुठलं?
ट्रिप्टोफॅन सिंथेसिस.
बर बर, ठीक आहे, चालेल. बघू आपण तू काय एक्झाम्पल घेते, आणि मग मी माझं एक्झाम्पल तर सांगणारच आहे, मग त्यात बघू आपण एक काय करायचं. ठीक आहे. सो आय थिंक विल स्टॉप हिअर टुडे. दॅट इज ऑल फॉर टुडे. पुट युअर रोल नंबर्स इन द चॅट बॉक्स. सोनम पाठव मला नंबर लिहून.
येस सर.
ठीक आहे. काही क्वेश्चन्स आजच्या याच्यात? You are good students, you never ask questions, but that is a very bad sign. Always you should keep your sessions interactive. ठीक आहे. तर माझ्या मते ह्या 2 ते 3 दिवसात आपला सिलेबस संपेल, आणि मग त्याच्यानंतर मी काही तुम्हाला तुमच्या टेस्ट घेणार आहे. सो रोज एक टेस्ट घेईल. तशी मी एक अर्ध्या तासाची टेस्ट असेल. So that will give an idea that what kind of questions could be asked in your final exams. शेवटी एक थोडक्यात मी तुम्हाला सांगेल की कसं अटेम्प्ट करायचं, काय करायचं, काय कशी प्रकारचे क्वेश्चन्स असतील वगैरे थोडक्यात सांगेल. and that will be all from my side as far as biochemistry is concerned. Right? So that's all. If no questions, we'll call it for a day. Good day all of you.